Embryology is the study of how organs develop in the human body. The fallopian tube is an important organ in female reproductive physiology. It helps transport eggs and is where fertilization happens. In this video, we will discuss how the fallopian tubes develop and their role in reproduction. The fallopian tube's mucosa contains ciliated cells that move eggs and embryos in a coordinated, directional flow towards the uterus. Non-ciliated secretory cells produce mucus, while smooth muscle contractions aid in the transport of eggs and embryos. Cilia in the fallopian tube not only assist in the transport of oocytes and embryos but also play a critical role in various physiological processes. The cilia within the fallopian tube are motile and possess intricate molecular structures that enable their coordinated movement. Defects in ciliary structure or function can lead to impaired fertilization and ectopic pregnancies. Research suggests that ciliary dysfunction may be associated with various genetic disorders affecting the fallopian tubes. Hormones play a pivotal role in the development and maintenance of the fallopian tubes. Estrogen and progesterone, produced by the ovaries, regulate the growth and differentiation of fallopian tube epithelial cells. These hormones also influence ciliary function and the expression of proteins involved in cell adhesion and transport. Any hormonal imbalance during development can lead to abnormalities in fallopian tube structure or function. Tubal ectopic pregnancy occurs when an embryo implants and develops outside the uterus, most commonly in the fallopian tube. Understanding the molecular mechanisms underlying tubal ectopic pregnancy is essential for developing effective diagnostic and therapeutic strategies. Recent studies have identified the involvement of various proteins and signaling pathways in the process of tubal ectopic implantation, including adhesion molecules, growth factors, and inflammatory mediators. The tubal fluid, secreted by the fallopian tube epithelium, comprises nutrients and signaling molecules required for successful fertilization. This fluid also aids in the maturation and capacitation of sperm, enhancing their ability to fertilize the oocyte. The composition and regulation of tubal fluid are influenced by hormonal factors and contribute significantly to the microenvironment necessary for successful fertilization. Implantation is a critical event in human reproduction, during which the embryo establishes contact with the uterine lining for further development. The fallopian tube and the uterus engage in extensive molecular and cellular interactions to facilitate successful implantation. Communication between these two structures involves the exchange of various factors, such as cytokines, growth factors, and hormones, which regulate embryo development and implantation. The fallopian tube can be subjected to damage due to infections, inflammation, or surgical interventions. Regenerative medicine offers promising strategies for repairing and restoring the normal structure and function of damaged fallopian tubes. Stem cell therapy, tissue engineering, and gene therapy approaches are being investigated to promote tissue regeneration and improve fertility outcomes. Genetic abnormalities can significantly impact the development and function of the fallopian tubes. Mutations in genes involved in ciliary structure or function, hormone signaling pathways, or reproductive development can lead to congenital anomalies or infertility. Understanding the genetic basis of such disorders is crucial for accurate diagnosis, genetic counseling, and potential therapeutic interventions. Epigenetic modifications, including DNA methylation, histone modifications, and non-coding RNA regulation, play a crucial role in determining gene expression patterns during fallopian tube development. Aberrations in epigenetic processes may contribute to fallopian tube disorders and infertility. Exploring the epigenetic landscape of the fallopian tubes can provide insights into the molecular mechanisms underlying their development and function. Comparative embryology studies the similarities and differences in the development of organs among different species. Comparative studies of fallopian tube development offer valuable insights into the evolutionary history and functional adaptations of this vital reproductive structure. By studying the embryonic development of the fallopian tubes across species, we can gather information about conserved molecular pathways and identify species-specific variations. The fallopian tube microenvironment plays a crucial role in oocyte and embryo development. Changes in the fallopian tube microenvironment, such as the presence of inflammation or oxidative stress, can negatively impact fertility outcomes. Understanding the factors that influence the microenvironment can help in the development of strategies for fertility preservation and enhancing assisted reproductive technologies. Organoids, three-dimensional structures derived from stem cells, have emerged as powerful tools for studying organ development and function. The development of fallopian tube organoids allows researchers to study the intricate cellular interactions, signaling pathways, and tissue architecture involved in fallopian tube development. These organoids can be utilized to investigate reproductive disorders, drug screening, and personalized medicine approaches. In conclusion, studying the development of fallopian tubes offers valuable insights into how the female reproductive system functions. Factors like transport mechanisms, cilia, hormones, genetics, and environment contribute to their proper functioning. This understanding is crucial for diagnosing and treating reproductive disorders, developing new treatments, and preserving fertility. Ongoing research in this area will improve our knowledge of reproductive biology and provide innovative solutions for reproductive health concerns.